So it's my pleasure to introduce the first panel of this afternoon, which is on big data. We alluded to it this morning, but there is an explosion of data, both at every level of the organism to behavioral data like our cell phone usage, that can be used if you're clever analytically, and that has its own challenges, to really understand patterns of disease and potentially to prevent them. So to lead this panel, our moderator is Caroline Buckee. She is an assistant professor of epidemiology at the Harvard Chan School of Public Health, where she's also the associate director of the Center for Communicable Disease Dynamics. Dr. Buckee's work at Harvard uses mathematical models to bridge the biological scales underlying malaria epidemiology. She works with experimental researchers to understand the molecular mechanisms within the host that underlie disease and infection, and uses both genomic and mobile phone data to link these individual level processes to understand population level patterns of transmission. Please join me in welcoming Dr. Buckee.
Thank you. So the world is changing very fast, as we all know. We've all seen those exponential graphs showing population growth. But population density is also rising very quickly. And as of 2014, more than half the world's population was living in an urban environment. And projections suggest that over the next 30 years, we will add 2.5 billion people to our urban environments, and 90% of those will be in Asia and Africa. So there's nothing a pathogen likes more than a densely populated slum. And I think that really highlights some of the threads that were brought up earlier today about the importance of embedding all of our research in the local populations and national control programs in low and middle income settings, and in particular for capacity training. So this session is on big data. Many of us don't really like the term big data, although it's catchy. So I would like to reframe this as a discussion about qualitatively new types of data stream that are often at scale, at the level of the population. And so yes, this requires new computational challenges, but I think by labeling things big data, we can miss the fact that all of these new data streams are embedded within traditional and small data that are absolutely essential. So epidemiological information on the ground, the importance of community healthcare workers, like was highlighted earlier when we discussed Ebola, really can't be overstated. So we do have many new opportunities. So I'm just going to give you some numbers to kind of frame how fast the pace of technology is changing. So it took on the order of months or even years to sequence a single human genome. In the year 2000, it would have cost hundreds of millions of dollars. Now it costs on the order of $1,000. And we saw this morning from Christian's talk that we can sequence viral genomes, which are much, much smaller than that, for $50, even less, over a very short time period. We also talked about CRISPR and the promise of gene-drive technologies for controlling and containing diseases. So we have this opportunity to really fundamentally rethink how we do diagnostics, surveillance, and track epidemics for containment. Mobile phones, which we all have now, except for some people don't have smartphones, but generally speaking, everyone has a phone now. So the World Bank estimates that in 2000, 12 out of 100 people had mobile phone subscriptions. Now it's 101 out of 100 people have mobile phone subscriptions. Clearly the distribution is not even, and some people have multiple subscriptions. But increasingly, cell phones are everywhere. And so not only does it produce what we call digital exhaust data, behavioral data about where everyone is, where they're going, what they're doing, it also creates a tool to engage directly with people and to educate them. Now, of course, what comes with that is also challenges. So we have this explosion of disinformation, the importance of rumors in containing epidemics and trying to reach people. And so I think it also brings up this question about where we aim our solutions. So designing the perfect app isn't going to work, clearly. We need to have cultural specific context when we think about engaging populations around epidemic containment and surveillance. So I think the challenges that we're going to discuss today that come along with the promises that big data provide are to do with the lack of analytics, the lack of validation, the need to integrate these new data sources with traditional epidemiology and national control programs. And then it also brings up some issues that are age-old problems for public health practitioners, like how do we balance individual rights to privacy, with public health? And I would be interested to hear your thoughts about that too. So our speakers, again, you have their bios. Our speakers are going to range from global health security and threats on a global scale that are represented by emerging diseases. And then we're going to hear about national control programs and how we can move them forward using new analytics and new technologies. And then lastly, Ami is going to discuss with us the promise of genomics for helping us understand human health as an ecosystem, an integrated system that reflects the dynamics of immune systems and our microbial communities, and what that means for developing new drugs and approaches to containment. So I will, without further ado, I will introduce our first speaker, Jim Wilson. Please join me in welcoming him.
Okay. So we're going to talk about something that is, I would say, a little bit off the beaten path of typical public health, and this is the world where public health intersects with national security. And I think this is an important topic to discuss because, and just for context here, this is the first time I've ever spoken about this publicly. And I've been doing this work for 25 years. Is what do I mean when I say health security intelligence? Why do we need this intelligence? I think your speakers from the first panel amply displayed why we need effective intelligence. How is information used to inform our intelligence? What do I mean when I say intelligence? I'm not interested in people's personal records at all. I'm not interested in named individuals. I'm interested in trends, big trends, scary trends, trends that are sometimes very hard to detect. How do we use the intelligence? And what are the challenges with health security intelligence in today's society? I will give you a hint. They are the exact same challenges that Kevin outlined earlier, as well as our first panel. This whole aspect of information sharing, who gets to control that information, all the games people play. You will find me to be a cynic. You will find me to be very frustrated and angry with my own career and my failure to convince our government and the world to do the right damn thing. Okay? All right, here we go. Traditional public health, and I'm going to upset a couple folks here, as a discipline does tend to be forensically oriented. There is an important reason for that, okay? You cannot have our CDC or our public health officials expressing things that are speculative to the public. You must have confidence, right? Are we all in agreement? You don't want to have questions about your CDC. You want to take their word as almost the word from some divinity, right? Okay, to achieve that level of confidence takes time. Takes a lot of time. Unfortunately for us, we continually run out of time. We don't have time to screw around. We need to make decisions now, okay? If I told you the real stories behind the scenes about how we managed Ebola, how we managed Zika, how we managed a lot of these issues, you know, and there are books out there like the band played on, you know, about HIV, you will find that we've been playing games for decades, and it's very unfortunate. But some of those games are important games to play. You need peer review, right? You do. You need to be sure of what you're dealing with here, okay? So there's a minimal tolerance of false alarms. In our world, however, we embrace false alarms. We want to be on that bleeding edge of alert notification. I am intolerant of people playing games with information when we're dealing with an event that could cause massive socioeconomic disruption or scare the living heck out of the public. I want resolution of uncertainty as fast as we can get it, and I'm willing to take false positives, okay? Now that's a very different perspective. It really is, in a totally different work culture. In my world, I was dealing with the time-sensitive issues, the very bleeding edge of a flip point where a community is just humming along in their activities of daily living and all of a sudden somebody flinches. So So the birth of health security intelligence really began, and there have been various groups that have looked at this from an intelligence perspective since going back as far as the Spanish flu of 1918. Okay, but they've done it for various reasons and with different purposes in mind. I actually got my career, or started my career, trying to forecast Ebola using satellite imagery. But my sample size was really small and I was having a hard time publishing the paper. Okay, because you want a robust sample size, right? You want to meet certain criteria for publication. We just couldn't get it out there. But I needed to get it out there because I needed people to test the hypothesis, right? So this is in the late 1990s, before we had BioRxiv and some of these other systems to enable us to share that kind of information. But when I walked around our country trying to find the one agency that actually has a watch cell that actually does indeed do nothing but watch the entire planet for bad things, I couldn't find it. And I say I went everywhere. I even went to the Department of Transportation in desperateness. Why don't we have a National Weather Service equivalent for infectious disease? I am angry. After 25 years, we still don't have it. So I tried to convince people, right? What would it take for us to have one for the world? West Nile virus was the beginning of that discourse. And I'm here to tell you that West Nile, unfortunately, sadly, did not kill enough people. You have to kill people. You have to kill Americans, you got to do it fast, and you got to do it in a nice cluster, and you got to get a good sample size, and you got to kill the young, and you got to kill working age adults. Think about it. What's it going to take to get political willpower to back something like that? What ultimately got us started was actually SARS, where our team actually looked at some of the signatures that were actually reported in publicly available Chinese media and realized that we actually missed plenty of heads-up warning for months. Okay, I'm sorry, the Chinese actually didn't really hide a lot. There was a lot of information, okay? Now, why did we miss it? Oh, there's some pretty ugly questions to be asked there, yes? 4.5 months is what I'm talking about in terms of the time delta. We've had alleged bioweapon events, including one that I recently published on the largest anthrax epidemic documented in modern history during the counterinsurgency in Rhodesia in the late 1970s. Interesting events that shouldn't have gone unnoticed, that shouldn't have gone unreported. Why aren't there many publications about these events? Lab accidents, we've had lab accidents, yes, many of which have not been reported. I think Kevin's provided us with ample hints of there's research going on around this world. Are we aware of who's doing what where? Genetic manipulation of H5N1 virus to get it to transmit more efficiently is my popular favorite. Do we know if they've had a lab accident? What happens if they do have a lab accident? Do we have vaccine ready on standby in case they do? Will it be able to be deployed fast enough? Unfortunately, these are the questions that we're stuck with in my world. HIV/AIDS, took us a long time to figure that one out. Venezuelan equine encephalitis, that's an agent that is mosquito transmitted, very similar to some of the other agents we've discussed today, causes pediatric encephalitis. We had a huge epizootic in Venezuela that, for which we didn't receive warning, and there was a convergence of the peak of the epidemic with the peak of direct nonstop air traffic to Dade County, Florida. West Nile, well, that's been robustly discussed by Tracy McNamara, who's the Bronx Zoo veterinarian who helped raise the alarm on that, and all of the challenges she faced trying to get people to pay attention to what's going on. And if you really talk to Tracy, you'll find out that later she had a really hard time getting a job. She is a national hero, in my opinion. She should have gotten a medal. SARS, we already talked about. MERS. When was the first time MERS was really detected? Wasn't in the Kingdom of Saudi Arabia. It was in Jordan. It too was blatantly apparent to any analyst who was monitoring in the native vernacular, right, or Arabic media, where they talked about an unusual, atypical, fatal illness killing ICU healthcare workers. Cholera, Haiti. I was asked not to reveal my suspicion that the UN Mirabai base was the source of the cholera disaster. My response to that was, if we believe the UN to be the epitome for the world, then act like it. It's called corporate responsibility. It was an accident. Do not deny it. Ebola. How long was it on the ground before we were finally able to mount a response? Chikungunya, Zika. This is a pattern that's been going on for decades. So targeting. So in our world, we really are focused on a subset of disease. The things that move very quickly are locally unfamiliar, right? So this speaks to diagnostics, availability of infrastructure on the ground. Has to be clinically significant, right? Can't be a mild disease. Ideally, it kills. Ideally, it maims. Ideally, it marks the individual so that any layperson can see what's going on. You have to affect a certain segment of the population, as I've hinted at before, children, working age adults. If it affects the elderly, unfortunately, that gets buried. People don't talk about illnesses in the elderly worldwide like they do with working age adults. Problems with access to diagnostics and countermeasures. So if we don't have a vaccine, that's concerning to us, right? If we don't have an effective antibiotic, efficient transmission is a big one, right? And that has to do with the dynamics. Do we have herd immunity on the ground or not? The data focus. We use global media. Now, I'm going to tell you flat out, no company on earth has a full grasp of the entire global output of media. If you think ProMed does it, guess again. If you think Google does it, guess again. Okay, Rupert Murdoch has played actually quite a strong role in actually restricting our access to media over time, because you have to pay for everything now. So now the costs have increased, right? We had to monitor, just to give you an idea, we had 50 full-time analysts who spoke 50 languages as their primary language. That's what it took to get at the barest tip of this iceberg, okay? We had 5,000 live events on the radar a day that we had to keep an eye on, 5,000 that were threaded, right? And you got to pick which one of those 5,000 events that you're actually going to pick up the phone and put your career on the line and call somebody about. Tough work. And human networks. And really what you're looking at is you're tipping and queuing. And it's a constant dialogue. It's a constant conversation. And you're having these conversations with researchers, with physicians, with journalists, with anyone who is close to the action to try to say, hey, is this something or is this nothing? At a time when we have no epidemiological data, at a time when we might not have any diagnostic information at all other than people are dying, or there's a report that people have panic evacuated a village. The bias, of course, when you're dealing with data of any kind in the electronic environment is, of course, where is their infrastructure to support that data, right? And so we knew very well that it tends to follow the city light map, right, which should make sense. It's where you have electricity, right? So in the analysis and dissemination piece, we have used forecasting tools for decades. Okay, and so if anybody tells you that we cannot forecast infectious disease, I'm here to tell you that's just a blatant lie. It ain't true. We do. We have been for a long time. Now, how do we use it? We use it to set the hypothesis when we're tracking something in the real-time space. We say, okay, I expect to see that, okay, we're about to enter cholera season. I'm also forecasting the entire social response pattern of that community. I know what they're going to do, what countermeasures they're going to deploy, how they talk about it in the media, whether or not there's social anxiety, whether or not the hospital gets overwhelmed. We have all that information, and we know what to expect. So therefore, if you know what to expect, you can recognize deviations, right? And you can recognize when, that train seems to be coming off the rails here. What's going on? Signal processing, I've kind of hinted to that. That's the recognition of an anomalous signature pattern or an anomalous pattern that's appearing in the system. There are a myriad of different ways to do that. We have pattern libraries. So there's an Ebola signature. And so one of our speakers from the morning was talking about the Nigerian experience. That signature looked like a country that is well adapted to handling those kinds of issues, whereas the signature of a country that has never encountered Ebola before with no infrastructure, it to us looks like a heart attack, wild fluctuations of indicators. Sensitization, desensitization of human networks. We will actually ramp up entire global networks of people to watch for something and then tamp them down again. Now, the tamping down is a really important piece that people really don't appreciate, is I spend actually 98% of my time calming people down. Indication warning methodology is really what we're talking about here, which is a Cold War intelligence analytic methodology, and it's available for you to review in the public literature. Now, here's an example. So we manage the Japan Medical Intelligence Center. These are actual forecasts for Legionella, carbapenem-resistant gram-negatives, which is a gram-negative bacteria that is resistant to one of our end-of-line categories of antibiotics. I'm also, side note, very much into antimicrobial resistance tracking. And EHEC, which is a disease we often see, or not often, but thank God not often, a disease we occasionally see in pediatrics. So the point here is, you know, I have a different demand for forecasting. I just want to know if we're about to hit the season or not, right? Across dozens of diseases, down to the prefecture level, down to the community level, right? And we have this information across multiple countries. It does exist. Here's for Nevada. And on the left side here, this is an example of an unusual fall outbreak of H3N2. And that's a seasonal influenza. And we were able to recognize it very quickly because we had the baselines for influenza. So a lot of this information is very reminiscent of what already happens in epidemiology, right? We know what the seasons are, right? And we do build statistical baselines. The question is so what? How do you interface with this information? How do you communicate with people? And how do you blend it with your awareness of what's happening in the community? And in this case on the left, what happened was physicians were recommending the influenza vaccine to older adults in December because of waning immunity. The vaccine protective period does not last as long in older adults. And so rightfully so, our community said, well, just wait till December to get that vaccine. Well, we had 4 fatalities due to type A influenza by mid-October that year. And it was actually a warning failure, okay? And I got in a world of trouble with my local public health people for pointing that out. RSV, you see that peak there, that red line goes above our forecast band. That's a point where I actually picked up the phone and called our pediatric intensive care unit to make sure that our sole PICU in the county wasn't getting overwhelmed. Okay? Are they calling up backup ventilators, this kind of thing. So it's very important to kind of put your data in context with your infrastructure. What's happening? Are we able to respond? Attribution. So this is the actual email that I sent to 1,000 people on the ground in Haiti about our suspicions about the Mirabelli base. I don't expect you to be able to read it. But the point here is that this was basically the culmination of this kind of messy world of data gathering and human conversation. This was the tip where we knew that we weren't dealing with endemic Haitian cholera, as some people were arguing, that we were actually dealing with an imported virgin soil epidemic of Nepalese cholera, whole different animal, right? And had national security implications from the Haitian government point of view. Signal recognition. So how did we provide the tip-off on swine flu? Well, that was open source media. Okay? Unfortunately for us, we ran into some crossed wires with CDC who didn't want to tell the press and acknowledge that we had given them this information. We lost a $10 million company over this. And I sent another 50 analysts home. Challenges. Well, as I've hinted, this is a direct challenge to established bureaucracies. There are many disagreements regarding what is considered an alert priority. I've spent my career playing Cassandra, the Greek tragedy of being cursed with the ability to forecast and no one will listen to you. Failure by bureaucracies to use the intelligence. I've seen this over and over and over again. I'm tired of seeing it. Lack of understanding what to do with the information. Well, okay, that's an important challenge, right, that perhaps I can help with. Information control. If you think that people don't control information, you're not living in reality. Everyone does it, and they do it in my own locality. Okay, I see hospitals hiding information all the time about drug resistance, for instance. And of course, denial deception. The funny thing is, if you ask anyone in the national security community, they will put that last bullet first, saying, well, the state actor is suppressing information. The hardest time I've had getting information has nothing to do with that. It has to do with my own local folks. Okay? So it's the Pogo cartoon, right? Who's the real enemy here? Challenges, academic and operations disconnect. So it's very hard to have somebody like me sit in on a collaborative research group to give them guidance and say, look, this is what's really important. If you go down this road, honestly, great paper, You get your PhD thesis, but we won't be able to put it to use. And there are all kinds of reasons for that. There's an operations policy disconnect. I hate this. This is the thing that actually is driving me squarely into insanity. Is we keep seeing policy drafted out of Washington, and it's quite clear that the people who drafted the policy have never done this work before. Okay? Transparent accountability. I've just hinted at multiple intelligence failures. When was the last time we had an open congressional session where we sort of reviewed who knew what when and why didn't you share that information? Time for that to change, I think. Substantial inability to provide balanced risk assessment. So this hints at our culture's tendency to hype things. Okay? I do think that we hype things. So I know I've got 30 seconds, so let's rip through this fast. Live example. There's been a proclamation that we're going to be hit with a severe influenza season. Some of you may have seen this, okay? And it came out of Australia. Australian media talking about the worst flu outbreak on record in 2017. Worst on record in 2017. In general, we get our season what the southern season what the southern hemisphere got in the season immediately preceding us. So this is Tony Fauci saying this. Now, if he understood perhaps what this looks like from our perspective, I wouldn't have gone that far out with that comment. I would have maybe hesitated to have said that. Because it's not actually very much a guarantee at all that we're going to see what they see in the southern hemisphere. But it hit CNN. Flu jab warning. NHS, National Health Service of the United Kingdom, warns of major outbreak. Getting vaccinated could be life-threatening. This is the United Kingdom. So there's a spread of a meme. British flu deaths may rise in the worst epidemic in 50 years after Australian strain outbreak. And now, the media is creative. They're going to swap that virus out and say it's due to H2N2, which was a pandemic strain not seen since 1957. Creative. I got to hand it to them. Again, current influenza season being worst on record, and evidence that the elderly are particularly at risk. So Sanofi Pasteur chiming in now to take profitability options here. And now the Canadians are picking up on this. Now Canada's all lit up, thinking they're about to get hit with a pandemic. This is all happening right now, by the way.
In the interest of time.
Yeah, it's going to be 2 slides. So admissions to Sentinel hospitals, right? So this is the actual report from Australia that started this entire train of events. Note the underlined comment, and not necessarily reflecting an increase in severity and infection. So what has happened here is that they're diagnosing more cases. It is not necessarily more severe, but don't let that stop the media. And then finally, we see somebody rational, after months of this spreading across multiple countries, this meme, finally somebody in Canada decides to say, no, wait a minute, maybe we're overblowing this. And then last but not least, what's happening now? We have perfectly routine influenza being documented in CDC, by CDC. Now, this may change, of course, okay? The influenza virus that's in circulation right now is covered by our current vaccine. So much ado about perhaps nothing. So I'll close with that, with the point that it would be nice to have a little bit of balance in the discourse.
Thank you. Thank you, Jim. So we can debate the how good forecasts are all day, but we can do that later maybe. Next speaker is Jess Metcalf. She's an assistant professor at Princeton. You can read her bio in your book, and she's going to talk about how we can move national control programs forward using modeling and new technologies.
Hi. So it's a great pleasure to be here. I'm going to talk about how we can actually connect some of these new, exciting, and, to use the term, big data streams to public health questions. And I'm going to focus my discussion around measles. So measles is a directly transmitted, completely immunizing infection. So that means for someone to give someone else measles, they have to be in proximity. And once you've had measles, you'll never have it again. So it has a very simple life cycle. Individuals tend to be born susceptible, they then become infected, and then recover and are protected for life. We have a safe, inexpensive, and completely immunizing vaccine. And since the case fatality rate for measles can be extremely high, as high as 20% in low-resource settings and in catastrophes. And because the consequences of measles can be so bad, measles vaccination has been referred to as a best buy in public health. Following successful eradication of smallpox and polio, the Pan American Health Organization decided to target measles elimination in 1994, a combination of routine vaccination with large campaigns designed to both interrupt transmission and maintain high population immunity meant that this was achieved within a decade. It's a very remarkable success. In the rest of the world, the situation was not so successful. So the x-axis on all these graphs is time, and the y-axis is numbers of cases. And you can see that red line is PAHO, Afro is the African WHO region, and WPR is the West Pacific region. They're falling much more slowly. Although there were substantial gains at the beginning, progress has been interrupted. And we know why, partly. There's considerable inequity in vaccination coverage across the globe. The yellow countries on this plot in around 2011 had coverage of less than 80%. Of course, these national statistics also conceal a lot of important heterogeneity, and even in countries that seem to be doing really well, there have been large, unexpected outbreaks. So this is an example of one in Malawi that had a well-performing system in very few cases for about a decade, and then experienced this large late-age outbreak in 2010. And it made sense. We know why that happens. Vaccination coverage was good but not perfect. So every year, a certain number of children were not being immunized, and they were also escaping infection because we had successfully reduced transmission. And that meant that when the infection came, there was this large outbreak. We used to think that measles was so transmissible, it is so transmissible, that there was no point trying to intervene once the epidemic had taken off. We know through careful modeling now that that's not the case. Outbreak reactive vaccination is now indicated by WHO. And of course, it's even better if you could have anticipated this outbreak. If 2 months or 3 months before the measles outbreak had kicked off in Malawi, you'd been able to reach the population and vaccinate it, you could have averted many, many cases potentially, and thus deaths and thus the morbidity. But what would we need to do that? We need two pieces to do that successfully. And the first is that we need to understand the landscape of immunity. We need to know who's protected from infection and who isn't. In this particular example, the person in red is infected, and I've set this up with an R0, so a number of new infections of around 2. So it infects 2 other people, but there's a third person who's immune, and as it progresses, and as the pathogen progresses, more of the population is protected. If we vaccinate effectively, everybody should have that little blue shield on them, and the pathogens should not be able to spread. To understand the landscape of immunity, we therefore need to understand both the scale of heterogeneity in local demographics, because the source of new susceptible individuals is births, but also the history of vaccination and infection. There's another piece that we need, and that is that we need to understand human mobility. So we need to understand if you have successfully eliminated measles in a particular location, but you are not vaccinating everybody so susceptibles can build up, you might still get away with it for a bit, because the only way measles is going to arrive in your city or your district is if someone with measles turns up there. So we need to understand how people are moving around the landscape in order to grapple with understanding the landscape of immunity and mobility to predict when outbreaks would occur. So what we wanted to do, and Caroline was part of this work, was see if we could figure out, retrospectively, using all the tools we had available, could we have got ahead of an example historical outbreak? And the example outbreak we used was one that happened in 2010 to 12 in Pakistan. So again, x-axis years, y-axis cases. So we mustered all the data we had available. And first of all, to calibrate the landscape of immunity, we pulled together the vaccination coverage surveys that had been done in Pakistan over previous years. And it's quite a luxury having vaccination coverage surveys because it doesn't exist for many of the settings where measles is of concern. We put this together with births, and we put this together with our best guess of what the history of infection had been. And this is actually a reasonable thing to do because in the era pre-vaccination, it's reasonable to assume that everybody got measles. There's this great quotation, measles is like love, everybody gets it. And it was definitely true back in the day. So we built a picture of what we thought the landscape of immunity was, and then to put this together with human mobility, we leveraged a source of data that Caroline really pioneered, which is using mobile phone call data records to capture how people are moving across the landscape. And the way this works is that every time you make a call, the tower that you're closest to routes that call. So if we know where the towers are, and we have a record of the calls, we can figure out these large-scale fluxes of population. And this data comes with no other information. We just know where people are and where they've moved to. We don't know their age. We don't know their vaccination status. What we have is fluxes of mobility. So to evaluate our ability to predict, we're also going to need information on measles itself. And so we had two sources of data here. One was province-scale case reports. So over the timescale from September 2012 on the left, going forward to September 2013 on the right, there were records of numbers of cases in each of the provinces of Pakistan shown by the colors. So we compiled that data. It's not perfect, right? If you're interested in human mobility, Pakistan's rather large, and provinces is rather a large scale to be thinking about this problem. So we also pulled in another source of data, and this was a different source of information, which was district levels alert data. So when local health officials of a particular district thought a measles outbreak was kicking off, that data was sent up to the Ministry of Health and was available to us. It's not actually instance data. We don't know the total number of cases that were there. They might just have flagged up that something was about to happen. Okay, so how am I going to see if I can predict this data? Well, what I'm going to do is build a model of what Pakistan looks like, and this is a very crude cartoon, obviously, for feasibility. If every one of the points is a different district, what the mobile phone data gives me is a sense of the fluxes of individuals between all these different points. So there might be strong connections between some districts and weaker connections between others. For every one of these districts, I also have a sense of what proportion of the population is immune to measles and what isn't, and that's going to matter for the pathogen spread. So I have my sense of how the world works based on all the big data that I have, and then what I'm going to do is initiate an outbreak in the setting where I know that the outbreak kicked off based on the district alert data. And I'm going to run a simulation. And this works essentially by saying the outbreak begins. It might have moved, if this was the district that was most connected, it might have moved here next. The outbreak continues in the two first districts and might move to a third, and so on. So over time, I'm going to have a full simulation of what the outbreak looked like in every one of the districts of Pakistan. And importantly, every time I rerun this, you might get a slightly different picture, because I'm assuming that chance is going to play a role, that some routes are going to be fairly similarly important at different times, and so we're flipping a coin to figure out which one predominates. So every simulation might end up looking a little bit different. But overall, they look a bit like this. So this is simulated trajectories across all districts in Pakistan based on the mobile phone data and the landscape of immunity data. The x-axis is time in bi-weeks over the time course we were focused on, and the y-axis is the number of cases we obtained. So we need a way to evaluate whether this is any good or not, obviously, and so for that we go back to our original district alert data. The province level data is just too coarse to do much with. And we chose to focus on the question of, could we anticipate, could we predict how many districts would occur after a certain point in the future, right? Because in that case, we could rush out there, we could start preparing campaigns, we could maybe get ahead of the wave. And that would be of considerable public health benefit. So on this plot, what we have is time in bi-weeks on the x-axis into the future, and on the y-axis is the proportion of districts that we accurately predict to have happened after the time point on the x-axis. Okay? So at the start of the outbreak, 100% of districts are happening in the future. We do quite well. That's not too impressive. And then the proportion predicted to happen in the future falls off. And by about 10 bi-weeks, so about 5 months, we're doing as well as a coin flip, right? So we're doing 50%. And after that, our predictions get even worse, because we have a model, and the model is less good than just saying stuff might happen by chance. So is it helpful? It's been estimated during measles outbreaks in Malawi that it takes about 2 to 3 months to get a campaign up and running. So we're buying a little bit of time there, potentially on that scale, but we're not doing great. And it's important to think about why we aren't doing great. And in this context, it's important to remember that every single source of our data, every one of our data sources has uncertainties in it. So the vaccination data might have reporting biases in it. We might only be finding the children to report on coverage who had been vaccinated, because the ones who aren't vaccinated, by definition, are probably hard to find. We might have a large population, therefore, of missed children. So our vaccination coverage estimates will be really optimistic. Mobility data, mobile phone data is really amazing and gives you this incredible window onto how people are moving around the landscape. We don't know if it's relevant to children. We don't know if it's relevant to transmission from one individual to the next, right? There's a quite specific biology underlying measles transmission, and these big fluxes might not capture it. And then the case data. It's true everywhere in the world. Even, an outbreak in Holland in 2007, it was estimated that 10% of measles cases were reported. Nobody reports measles cases. So that data is also fairly erratic. And I think this comes back to a thread that Caroline alluded to in the beginning. To make any progress around really leveraging these unique and powerful novel sources of information requires really strengthening existing sources of information, really basic epidemiology, case reports, vaccination coverage. No one ever seems to write down what fraction of the population they vaccinated, possibly because they have more important things to do, like vaccinate children, but it would be helpful. So one of the things we've been thinking about as we move forwards in this space is I told you I need two pieces to really try and predict where and when an outbreak is going to happen. And even for measles, which is perhaps one of the world's simplest and best understood of pathogens, it's really hard. And we ended up thinking about, is there another way, is there another approach we could take to refining our understanding of the landscape of immunity? And really, we started from the fact that this is the central process in epidemiology, right? I told you individuals start off susceptible, that's the S. They become infected if they encounter an infected person, that's the I, and then they recover. And just about every piece of modeling around infectious diseases has some variant of this basic schematic in it. Yet all we ever see when we're trying to build these forecasts is cases. The susceptibles and the recovereds are invisible to us. And we can infer who's susceptible and who isn't, right? That's what I did here. I pulled together information on births and vaccination to try and figure out who could have got the infection, who was protected and who wasn't. And in measles, this is an example of pre-vaccination measles in England and Wales. It's very straightforward. The number of susceptibles in 2 weeks' time is the number of susceptibles now, plus new births, minus whoever got infected. So you just iterate it forwards, and you have your trajectory of susceptibles. Almost everywhere in the world will be a lot like Pakistan, where the case data is shaky, the vaccination coverage data is shaky. Everywhere now has measles vaccine, so you have to account for it. But it turns out that there is potentially another approach to getting at that, and that is that serology can directly measure susceptibility. So this is where if I were to go out into the audience and take your blood, which would be very strange, I could evaluate whether you had been vaccinated against measles or not. And I'm guessing that everybody in this audience has an antibody titer that is protective to measles. In my case, it's actually because I was infected, but we can discuss that with my mother. And you can do much more nuanced, really interesting things. So for example, I could evaluate whether you'd been exposed to particular flu strains, like Hong Kong 68 or Bangkok 71, which tells me whether you were in a part of the world that was connected to the rest of the globe during these global pandemics. So it's an incredible window onto the past of populations, and it's an incredible window into predicting the future and the likely burden of infections. It allows us to project, it gives us a direct measure of this landscape of immunity. And so with colleagues, one of the things we're very excited about moving forwards in considering big data and the world of contagion and epidemics is developing a global immunological observatory. And our logic here is that if you think about weather forecasting, we have buoys distributed all over the seas of the world to measure sea surface temperature. to strengthen our prediction of global climate. And yet we have nothing equivalent for infectious diseases. But all of our blood is this unbelievable sensor of all the things we've been exposed to, all the things we've been infected by. And the technology is ramping up to a stage where you need smaller and smaller samples, and you can get more and more resolution across ever more pathogens. It would give you the prospects of anticipating hidden epidemics, or at least understanding when they were occurring, long before perhaps cases blipped up on the radar. So you might be thinking, well, that's not, how are we going to store that? What are the ethics of it? And it's absolutely true. There's a huge amount of both ethical and logistical considerations that have to come through. But I think the prospects are such that there are so many ways in which you could use convenient samples. If you think about blood banks, we could be using blood banks to think about what the world's kind of epidemiological and contagion risks are. There are all sorts of carefully designed nationally representative surveys that could add a biological sample to it. And there's even the prospect of not needing to use blood, because it's really hard to get at blood, but using saliva for some of these techniques with many of the new technological innovations, many of which are actually happening at Harvard, that would allow us to do this. So that's all I have. And I did all of this with Caroline, Andy Tatum, and other co-authors listed there. So thank you very much.
Thank you, Jess. So our final speaker for today's panel is going to move from, we've been to global threats and national vaccination programs and serology. Ami Bhatt from Stanford is going to talk now about a more personal scale, thinking about the microbiome and how our bacterial communities and viral communities interact to create susceptibility to disease and what it can teach us about the development of new drugs and approaches to make people healthy.
All right, so thank you very much. What a pleasure to be a part of this event and to be a part of this panel. So I'm going to speak for the microbes today, and I'm going to think about epidemics with you all through the lens of the microbiome. Whether we like it or not, a huge wave of data are coming at us, and we're just at the beginning of trying to build tools to understand these microbes, to enumerate these microbes, and to figure out how they change over time and space. So I'm going to start out by helping you get to know your trillions of neighbors. I'll try to convince you that context matters, and I'll try to give you a sense of the perils of a simplified microbiome. I'll scare you a little bit. Our microbiomes are under siege. And I'll remind you that while we talk about pathogens and microorganisms as though they're stagnant organisms, they're alive and they're changing. I'll end by giving you a very simple example of microbial evolution on a clinical timescale. So number one, getting to know your trillions of neighbors. You know, we are one, but we are many. There are about 10 to 100 trillion microbes within each and every one of us. They make up about 3% of our human body mass. We have 1 to 10 times as many microbes as we do human cells. There are 10 to 100 times as many microbial genes as there are human genes. And of course, as you all know, the largest number of microbes that we have are in our GI tract. Over the course of today's seminar, we've heard a lot about how microbes can be bad. And I'm not here to disagree. But sometimes they can be good or neutral. We know that we exist or coexist with these trillions of microorganisms and that they are inexplicably intertwined with ourselves. And so while many of us will look at this picture and see the host or the faces, when I look at this, I see the microbes. And what I understand is that if the microbes change, well, perhaps we will as well. And so part of what motivates me as a physician scientist is trying to understand this interface so that we can manipulate host health through manipulation of the microbiota. So I alluded to the fact that microorganisms can be good. And to make this point, I'll actually pull an example from the animal kingdom. I like elephants, which is part of why I have this picture here. But it turns out that this is also an excellent example of why microorganisms are good. So Elephants are one of many organisms that actually eat poop. They don't eat poop throughout their lifetime. They only do it when they're children. And if babies do not eat stool from adults from the herd, they actually have a runting problem, which means that they don't grow properly. The microorganisms that they acquire through adult feces actually help them to extract energy from their food optimally. So this is an excellent example, I think, from the natural world about microbial good guys. When we think about microbiome sciences and the little tidbits that I gave you earlier, a lot of how we've enumerated and understood the microbiome has relied upon techniques for enumeration. And while light microscopy was probably the first method that was successfully applied to enumerate microorganisms that were visible, there are other methods as well. And a vast majority of these methods now rely upon DNA or RNA sequencing. So Professor Happy earlier today talked about beautiful work using DNA and RNA sequencing to enumerate microorganisms, in that case viruses, and to understand not only whether or not they exist within a human, but how they change over time. And so the fundamental basis of most of the experiments that I'll describe is taking a sample, usually a mixed sample, sequencing it, generating these lists of A's and T's and G's and C's, and then trying to infer from that information what microorganisms are present. Put more simplistically, my job is to collect stool, to extract nucleic acids, and to ask simple questions like, who's there? What are they doing? And what do they make? In order to do so, obviously, we're generating gigabytes and now terabytes of data, and so we require high throughput computational pipelines to actually achieve this. So next, we're going to talk about context. And I'm going to try to convince you that context matters and that having a simplified microbiome comes with its problems. So for that, I was really happy to see that I have this orange. I got this orange for my lunch. I'll eat it at the break. This is an orange. This is probably not very scary to any of you. I presume you do not find it threatening. You probably also do not find this orange threatening. I think almost all of you have seen this movie. This is from The Godfather. For those of you who haven't seen The Godfather, you should. These are an excellent series of movies. But on a more serious note, in The Godfather, you may notice that every time there is an orange in a scene, it actually portends bad things. And usually the person engaged with the orange dies. Sorry, spoiler alert. So, these oranges, not so bad. Those oranges, quite worrisome. And so an orange is not an orange is not an orange. Likewise, microbes are not always pathogenic in all circumstances. And so the context in which you view those microbes and the context in which they exist actually strongly affects their function. So we'll turn now to the perils of the simplified microbiome and think about context in that way. So here are two microbiomes. The one on the left is a complex microbiome. This is what, for example, your stool microbiome might look like. The one on the right is obviously very simplified. All you see is these good and plenty looking rods. It turns out this exists in clinical medicine. So an example of how this exists in clinical medicine is in the setting of C. diff. Many of you have heard of C. diff, Clostridium difficile. Now actually it's called Pepto-Clostridium difficile, is an organism that is an opportunist. And typically, it exists, colonizes many healthy people. We have a few hundred people in here, about 5% of us are colonized with C. diff. Doesn't cause any problems. However, in the setting of a simplified microbiome, when you don't have a healthy context, it causes disease. So this is an example of an opportunist that is pathogenic only in a given context. And so while we can look at pathogens and infectious disease and epidemics and contagions through the lens of the pathogen, we can also think about it from the standpoint of those who are susceptible, as Jess pointed out. Now, many of you know one of the most successful treatments for C. diff is actually not antibiotics, but fecal microbiota transplantation. This happens to be my favorite picture of FMT, which shows you connecting two people's colons, which we obviously do not do. Over on the right, this is probably not right after lunch picture, but this is an example of a crapsule. This is just from across the river over at MGH. A colleague of mine, Libby Homan, generates these crapsules. And actually, if you take 15 of those pills one day, 15 pills the next day, you can actually have a very high rate of cure of your C. difficile. In part because what you're doing is you're giving a healthy microbiome context back to an individual who has a simplified microbiome. How cool is that? Now, of course, our microbes are under siege. I'm worried about them. I know you guys did a good job of going and having lunch and taking care of your microbes. But many have argued that industrialization increases exposure to agents that change our microbiome. So when you reflect upon your daily life, what are the things that we're doing that might simplify our microbiome? Well, if we look at humans throughout evolutionary time, early on in the hunter-gatherer days, the main thing that affected our microbiome was our diet. Our diet and occasional exposure to pathogens that caused terrible diarrhea and essentially kind of a clean out. As we went to a more agricultural sort of setting, diet again was a major effector. As we became more and more industrialized, heavy metals and arsenic were introduced into our environment. And finally, in the modern day, adding on to those heavy metals and arsenic, we also have broad-scale exposure to antiseptics and antibiotics. So by a show of hands, how many of you think you might have been exposed to antibiotics today? I'm going to argue that it's everyone. Okay. So this compound over on the left is triclosan. Triclosan is a common antiseptic. It actually was originally developed in the 1960s as a surgical antiseptic. It's a very simple molecule, and for those of you who aren't chemists, anytime you see a simple molecule, you can look smart at a cocktail party by saying, I bet somebody synthesized that. Because when nature synthesizes molecules, they're awfully complex. So this is a synthesized antimicrobial. It's very broad spectrum, and it's in thousands of consumer products. Now, you may know that the FDA banned triclosan and several related compounds in 2016. That ban just went into effect. So you won't see antimicrobial hand soaps on your shelves, for example. Now, while it was banned in certain household wash products, it's still present in lots of things. And I would guess that even the chairs that we're sitting on today have antimicrobials that are impregnated into the cloth. So Jessica Robato, a talented graduate student in my lab, decided to study this, and we collaborated with an epidemiologist named Julie Parsonet at Stanford to evaluate the impacts of triclosan on a cohort of mothers and their babies. To make a long story short, this is a complicated graph, but suffice it to say that each dot represents a bacterial taxon, so a community of different similar types of microbes. If you have dots to the right of the center line, these are microbes that are enriched in households exposed to triclosan. If you see a dot on the left, those are microbes that are enriched in households exposed to the control agents. And what you can see is in these adults, they happen to be mothers, over the course of time, we see an enrichment of these blue dots on the right side of the graph, which means these are organisms that are enriched in households exposed to triclosan. Interestingly, in this experiment, what was done was between 2 and 6 months, these households were randomized to be exposed to toothpaste that contained triclosan. Now, triclosan is not banned from toothpaste, so while you won't see it in antibacterial hand soaps, you can walk down the street to CVS and buy this particular toothpaste. And you can see that this is actually having a broad effect. Triclosan is actually the second most abundant human-made chemical in wastewater treatment plants, and it abounds in our groundwater. So whether we like it or not, we're exposed to this compound, and at least these data suggest that these compounds are changing our microbiomes. You can look at this in Jessica's paper that just came out if you're interested, and I'm certainly happy to answer any other questions you may have on it. But remember, not only are our microbiomes under siege and our microbiomes are changing, but our microbes themselves are alive and changing. They adapt beautifully to stressors. You saw evidence of how they adapt over time and acquire mutations in the setting of outbreaks. Fundamentally, for those of you who are not molecular biologists or geneticists in the audience, I thought I would just break it down into two main categories of ways microbes evolve. They acquire small mutations, and so in this case, the highlighted red letters are changing to another letter. or they can acquire genes or gene fragments. And we usually call this horizontal gene transfer. And so there are two main ways bacteria can evolve. It turns out that we're pretty good at measuring the way that organisms evolve on the left. So we can measure those mutations. Those are pretty easy using current technologies. But measuring the thing on the right has been difficult. And part of the reason it's difficult is because when you have a horizontally transferred segment, what it means is that what you're seeing in this toy example actually occurs, which is you have a repeated segment, in this case labeled R, that exists both between A and D in this case, as well as between B and C. Now this happens in nature, horizontal gene transfer. An example of where it happens is in this organism, E. coli. So I'll just ask you to raise your hands. How many, when I say like E. coli, feel really good about E. coli? That's like a few. How many of you feel like kind of bad? Like we're at a pathogen conference, so yeah, many of you feel bad. And how many of you feel like it's complicated? Yeah, like I feel like it's complicated. You know, on one hand, it's like the most commonly used bacterial model organism. Every biology lab in this country and probably in the world uses this. On the other hand, it can cause enterohemorrhagic E. coli, which we heard about earlier today, terrible diseases, kill people. And it turns out that part of the reason we feel conflicted is because E. coli can acquire genes through horizontal gene transfer that make it very virulent and very dangerous. Okay, so once again, E. coli is not E. coli is not E. coli. So microbes can certainly evolve. We know that. We know microbes can acquire genes through horizontal gene transfer. We've talked about how it's a little difficult to measure. But one of the things that we realize is that not only can microbes evolve over timescales of tens of years or hundreds of years or millennia, but they can actually evolve in real time. And in order to evaluate that, our laboratory studies patients undergoing bone marrow transplantation. So bone marrow transplantation is a potentially curative therapy for patients with refractory malignancies, so cancers like leukemia and lymphoma. It's a really intensive treatment, and it has a very high complication rate. But to take you through the general process, we have a patient. We give them a lot of high-dose chemotherapy. Often we'll also give them radiation therapy. This renders them much reduced in their cancer burden, but it also means that all of their rapidly dividing cells, including their red blood cells, white blood cells, and platelets, basically poop out for a period of time. In order to rescue them, we give them hematopoietic stem cells that are obtained from a healthy donor. And classically, we obtain these from the pelvis. And so that's why it's called a bone marrow transplant, because we took the marrow out of the bone. We give these stem cells to the recipient, and then we sit around and wait while a whole new immune system develops, which is kind of amazing. But as you can imagine, over the course of this time, these patients are very immunosuppressed. They're exposed to a lot of potential antibiotics, et cetera, to try to both prevent them from getting infections and to empirically treat them when they get things like fevers. Because they're exposed to so many of these compounds, their microbiomes are for sure under siege. And this is an example of a hospital patient at Stanford who underwent bone marrow transplant or hematopoietic cell transplantation. These are time points after transplantation. So A is before the BMT, and then B, C, and D are after. And what you can see is their microorganisms basically go away. This is a stacked bar histogram, relative abundance on the y-axis. And you can see that that blue organism seems to take over. And that blue organism happens to be Bacteroides cacchi. And so two talented graduate students in my lab, Alex Bashara and Eli Moss, asked this question, well, how does this occur? Why does that organism win? It must be particularly fit. Furthermore, they said, well, maybe gene transfer or gene movement makes this organism particularly evolutionarily fit. And they knew that measuring this gene transfer was difficult. Part of the reason it's difficult is because if we simplify that toy diagram into another representation, what you can see here is that we can't figure out whether the right path is ARC or ARD, BRC or BRD. What they decided to do was to use a technology that actually was commercially available, but no computational approach existed to apply this technology in microbiomes. And what this technology roughly does is allows you to trace a trail through the right path. In so doing, they found some intriguing results, which I'll share with you. I'm going to show you the genome, or a piece of the genome of this organism in blue. This is what a regular assembly looks like. This is 1/6 of the genome, and what you can see for those different time points, A, B, C, and D, is you have a lot of holes. So it's not contiguous. When they applied their new method, they ended up with these blue bars, so a much more contiguous genome. What this allowed them to do was the following. They were able to identify this huge segment at 60 kilobase pairs of DNA that actually popped into the genome between time point C and time point D. Right, this is horizontal gene transfer in a clinical time scale. This happened over the course of days and weeks, not months. Okay, so suddenly we can measure genes being transferred between organisms. Finally, they were able to identify insertion sequences, or small sequences that can jump around a genome, so jumping genes, as we heard about earlier. This particular jumping gene was absent in time points A and B and present in time points C and D. And these are the results. You can see I've got time points B, C, and D here. The gene ends up in that center, which is listed as the IS. And you can see what happens to the right. To the right of that gene, what you see is the genes just upstream are turned on. Okay, so whereas in time point B, you don't see a lot of expression of that gene, those genes are off. In time point C and D, once that insertion sequence pops in, suddenly you've flipped a switch and this antibiotic resistance gene turns on. So no genes were acquired or lost. This jumping gene just moved around and acts as a random light switch. So I'll just end by reminding you, we have trillions of neighbors, and our microbes are often good. Context matters, and there are perils associated with simplified microbiomes. They may increase our disease susceptibility. Our microbes are under siege. Antiseptics, antibiotics, industrialization, and many have argued simplified diets, low in fiber. Our microbes are alive and changing, and they are steadily evolving through mutations and gene transfer. Finally, microbial evolution can happen on a clinical timescale. I showed you evidence of a shape-shifting genome and antibiotic resistance. So with that, I'll thank my lab, thank all of you, and I'm happy to join the panel for some questions.
Thank you.
I was reaching for an orange microbiome.
Okay, thank you to our speakers. I think this means that we have 6 minutes of questioning between us, and then we'll open it up to the floor. So those were fantastic, and I think they really spanned a huge spectrum. I guess since we just ended there, Ami, I will ask you. There's this huge amount of data that we can now get about our microbiome. It's sometimes hard to make sense of which bits of it are epidemiologically meaningful. Can you imagine a scenario where, we've got Jess's serological bank, and we could have a similar microbiome bank where we think about vulnerability? Because ultimately what we're trying to do is think about who's susceptible to disease of various kinds. And I wonder if you can comment on that and on the current work being done in low-income settings on this type of microbiome simplification work.
Absolutely. So excellent questions. I think fundamentally, our microbiomes are great reflections of how we experience our environment. I'll take your last question first, which is this question of studies in low and middle income countries and countries that are currently undergoing rapid industrialization. There have been very limited studies on the microbiome of individuals outside of high-income countries, and indeed outside of the US and Western Europe. So this is an area of much need. We don't have adequate reference databases that tell us what the microbiomes of the vast majority of people in the world look like. The studies that have been done have pretty much been done in small tribes, often that are living in subsistence farming communities or in hunter-gatherer communities. There, it's been demonstrated that, at least at the simplistic taxonomic level, meaning who's there, the microbiomes are quite a bit more complex than our own, suggesting that Western diet, the Happy Meal lifestyle, may be compromising the complexity of our microbiomes. To your point about, will we ever get to the point where we can measure microbiomes and actually be able to predict disease susceptibility? I think we can absolutely get there. I think we need to do two things to achieve that. in addition to collecting data from countries outside of those where our data are overrepresented. One is collecting very high quality clinical phenotype data. So whenever we collect someone's stool sample, it's absolutely critical that we collect information about, for example, serology, disease history, migration, lifestyle. Secondarily, I think we need to be committed to measuring what matters. And in our field, we've been very reliant upon measuring taxonomy, meaning who's there, which genus, which species. But as I've showed you, these genomes can evolve. And so maybe we need to think more about measuring elements of the microbiota that actually lead to functions and measuring what matters. Maybe we need to measure what these microbes make or how they're changing over time.
Right, thank you. Or think about antimicrobial resistance plasmids and things like this. So Jess, I'll move to you. So, and also you guys are welcome to ask each other questions, but I was thinking about your serology bank, and we think about surveillance for emerging epidemics and zoonoses, and there are many who advocate that we should be going out and sampling macaques and the wildlife populations. Is there a way to use serology data to try and understand vulnerability to emerging pathogens that we have not yet seen? Do you think that's reasonable, or do you think it's limited to things that we already understand?
I think that it's challenging to look for what you don't know. But I do think that once an emergence is suspected, that's where the power of serology might come into play, because quite often many of the pathogens, we heard a lot about this morning, many pathogens can have asymptomatic infections, and so you might have no idea of the scope, particularly given uncertainty in reporting systems. But if you happen to have a blood bank, in northern Brazil or other locations where you suspected circulation, you would have a way to ground truth that and a way to understand what might be going on.
Yeah, and for Ebola too.
And for Ebola too. I think there's a lot of interesting questions about, whether our evolutionary ecology theory could actually take us towards thinking a little bit more about what would emerge where, but I think that's at a very early stage in the science still.
Thank you. And then I'll ask a question of Jim that's maybe different, but I think a lot of the data sources that you talked about are often in the private sphere, and corporations own the data and are differentially regulated or have different incentives. What do you think the role of corporations is as we move towards preparing for pandemics?
Well, that's a loaded question. I did disclose here, and again, this was the first time I've ever said this publicly, but we lost a $10 million company doing the right thing for the world. What incentive does anyone have and this has been mentioned before by other speakers, what incentive do we have to share information when you face that kind of a penalty? You blow your market. If I make the information available for free, and part of what killed the company is that we actually released the output of 2 operation centers online on Twitter. After we notified CDC, I told the operative, I said, listen, I'm going to give you 24 hours to get this out there to the American public before we go live with everything we've got. And we did. But what it did was it blew our market because you devalue the product. Okay, so there was also questions asked earlier this morning about vaccine. What's the status of the vaccine? Well, if you've noticed, we keep getting surprised over and over and over and over and over and over again, right, by these crises that are very time sensitive, and then we see somebody go before Congress and asking for $1 billion to develop a vaccine, right? Well, then all the information that's associated with that endeavor starts getting clamped down and becoming proprietary, and we had huge trouble getting information on what was happening with Ebola on the ground as well as Zika because of some of the overlays that were involved with that process, right? And I would ask the audience the honest to goodness question, do we have a vaccine available on the market for Ebola? Where did that funding go? Why don't we have a vaccine for Ebola? Why don't we have one for West Nile? Why don't we have one for SARS? Why don't we have one for MERS? Why don't we have one, so if you think about this, and Sanofi backed out of the Zika vaccine production, right? So where are we going with all of this? So allow me to be the curmudgeon here and maybe the cynic, but we have real problems with how do we share information and do the right thing. And that's why I'm very animated about this issue, is that I've seen it play out over and over and over again.
Yeah.
So to me, that is the big research question.
Okay. Yeah, I agree. There's an organization called CEPI that was founded after the Ebola outbreak, and it's an effort to finance clinical trials to get some of these potentially emerging pandemic infections, to develop vaccines for them, and so that if something happens, there's an accelerated pipeline to get vaccines out. It's a hard problem to know which pathogens you choose and all of those things, but it's certainly something we're going to need to tackle. Okay, so I'm going to open it up. If you come and line up behind the microphone, please state your name and then ask your question. And we have about 10 minutes for questions.
Paul Benninger from Tufts. Jim, I'd like to ask you about a whole half of the, maybe more of the environment that wasn't talked about, and that's the zoonoses. There's, we aren't coupling with the veterinary community. We're not with the zoos or with all the, the animal side of the kingdom here. Subhuman, blah, 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 blah. It seems to me that part of what you're doing really needs to be including that part as well. For example, the Journal of Emerging Diseases from the CDC has been doing that for the last 10 or 20 years in terms of making those connections. Any comments on that?
Do you want me to respond now?
Or should we, let's have a few questions and then we'll answer them.
Sort of a complicated question. My name is Alyssa Goodman, and I'm a professor in the astronomy department and also the other science director here at Radcliffe. And my question actually sort of concerns astronomy. So in astronomy, we're known for sharing our economically valueless data very well, actually. And so I was asked a few years ago to give a talk at something called SAGE Bionetworks, which is where a number of the pharmaceutical companies, thanks to the efforts of Stephen Friend, have volunteered to give away the data that were not useful in drug trials. And it turns out that this is really helpful. And so I'm just curious whether in the world of epidemiology, there's some equivalent of that, where some fraction of the data could be made public rather than all or nothing.
Thank you.
I'd like to thank the panel for your wonderful talks. Specifically, I'd like to ask about sort of what I think is an elephant in the room. I loved Professor Bhatt's, Dr. Bhatt's picture of the elephants. And that elephant results from the intersection of genetic engineering and perhaps particularly CRISPR, or CRISPR speaker talked about supervillains. And so my elephant is the intersection of genetic engineering, particularly maybe CRISPR-powered genetic engineering and bioterrorism, and how that relates to your interests in monitoring and responding to health crises.
I'm Ann Patterson. I work on an organic vegetable farm, which shows my bias. Can you give any guidance on how to improve our microbiome by what we eat? I know you said it was in the groundwater, but there must be something we can do about it.
Okay, thank you. So we'll start with the question for Jim about zoonosis and the importance of monitoring, being involved with agriculture and zoos and this kind of thing.
So, so Tracy McNamara from West Nile virus days, the Bronx Zoo, she was the one, way back when, who introduced me to the concept of One Health. So we definitely do. I was remiss in mentioning that, so thanks for calling that out. When we led the tracking of H5N1 when it was spreading throughout Asia, one of the leading indicators that we knew that H5N1 was probably spreading up into Russia was the bird die-off reports, right? So we're absolutely on that page and on board with that. And honestly, huge parts of the network that I monitor on the human side are veterinarians and are veterinary researchers. So we're very close to that. So thank you for calling that out. Yeah.
So the next question was about giving away seemingly non-useful data and whether we could do a better job. I mean, I think we definitely can do a better job, and there needs to be better incentives and the responsibility by researchers, like we saw earlier today, to take it upon themselves to do a better job with this. It's very hard as an academic because your incentives are squarely in publishing. And even if it's not for you, you want your PhD students and postdocs to get their publications out. Jess, do you have opinions about, I know you have opinions about this, so I will ask you.
So I have one lovely example of there's a chap called Hugo Lopez-Gatell, who was a PhD student at Johns Hopkins and went to the director of epidemiology in Mexico and asked for their childhood infection and other data. And the director of epidemiology told him no. And later, Hugo Lopez-Gatell became the director of epidemiology in Mexico. And all that data is freely available online. And one of my brilliant ex-graduate students, Aisha Mahmood, who's in the audience here, I think, used all that data for one of her chapters of her dissertation. I think it's increasingly moving. The Pan American Health Organization are real leaders in this respect. So many of those countries share so much of their data. And many countries are relatively relaxed about sharing their death records, causes of deaths, and so there's incredibly rich data there, which has yet to be mined. I'm the optimist. I think that the world is moving towards this, and the generation of scientists that's being trained now is thinking deeply about these questions and tackling these questions.
And I think digital platforms for data sharing are going to be key, especially for the kind of field epidemiology that we've been talking about. Once things are digitized, we don't have to go and find somebody's hand-drawn map, and things can be collated and aggregated and shared more easily. Do you have comments on this, Ami?
I think it's critically important that those data are shared, but I also think we need to support the centralized resources that host the data sharing. So from our perspective, we have open sharing mechanisms and approaches for genomic data, but our data are increasingly growing in size and are only accelerating. Right now in the United States, the National Library of Medicine is charged with storing all of these data, but are quite chronically underfunded for that purpose. And so I think as we think about generating these data, which we most certainly must and should share, we need to think about how we create infrastructures that allow that.
Great. On the question of genetic engineering and bioterrorism, I will pass this to Jim in a second. I would just say that like our ability to prepare for pandemics globally and our ability to detect things that are going to become a big problem, I think the answer often is health system strengthening, good access to healthcare in low and middle income settings. And it comes back to that over and over again. We can't detect anything if we don't have good data from the field. But I'm not an expert in the bioterrorism field, so I'll ask Jim if you'd like to comment on that one.
So the way that we detect unusual events is we're just looking for unusual disease, okay, or unusual events that are reported, unusual social disruption, right, where we don't expect it. And then we ask the questions, and then it becomes very forensic, right? The hard part here is putting things in context, and we have a habit in this country, and goes back for decades now, of worrying about biotechnology, right? So for those of you who are old enough in the room, you remember the concern about DNA recombinant technology in the 70s and the Andromeda strain movie and how it got everybody spun up about having physical containment level 4 facilities be created here in the United States. So this is a long-standing thing, and then we have Hollywood driving the memes, right? So we love to scare ourselves. We do. It's our society, right? And so if you watch these events, though, for 25 years and you become a curmudgeon like me, you realize that, we tend to blow things out of proportion a lot, right? So earlier we had a map posted on Zika that highlighted literally half the United States at risk for Zika. And we had to spend a lot of time around behind the scenes calming people down and saying, listen, the probability of a sustained epidemic of Zika is, okay, let's be cool about this, all right? Let's be careful. Let's put things in context. So I think we do have, when it comes to CRISPR, I think we need to slow down. Let's listen to the researchers. Let's listen and give them an opportunity to engage in the discourse. And let's not go a little overboard, as we classically do. My last point would be, we can't even handle game playing level 1. Why are we trying to jack it up to game playing level number 50? Okay, so we're having a very hard time managing the things that are presented to us by Mother Nature. Why do we have to go off into science fiction land? Okay, so I'm sorry for taking the pragmatic point of view, but that's really, after 25 years, that's what it's left me.
Yeah, and I think preparing for a pandemic means preparing for local epidemics first. So pandemics start local, and we have to think about it in that context. So the last question, and then if we have time, we'll take a few more, was about microbiome and diet. That was for Ami.
Yeah, so I think, obviously we're interested in improving our microbiome, and I'm glad at least there are some people in the audience who resonated with that. There's limited prospective data on this topic. Certainly many espouse eating diverse, fiber-rich diet as a method for potentially increasing or returning to an increased diversity of the microbiome. I certainly think as a physician, minimizing exposure to unnecessary antibiotics is critical. And I've seen certainly a shift from, 5 years ago when you have sniffles, you go in, you ask for your Z-Pack, to people coming in and really saying, well, I don't think I need a Z-Pack, which I think is part of the answer. Obviously, we have antibiotic use that's, broad scale in, for example, the livestock industry, and you heard a little bit about other household antimicrobials. Curtailing use of these is much more of a global issue than a local issue, but I think it begins with consumers asking what the potential for harm is with novel agents that are introduced into the commercial sector. And so I think being smart consumers can certainly contribute. But I think those are some of the things that we can do as we consider our microbiome. And I think the first step toward improvement is obviously awareness.
Okay, I've been told we have time for one more question.
Cynthia Chample from Brandeis University. And I was wondering, I think maybe this question is mostly for Jim, but are you doing any work with tuberculosis? Because it's the leading infectious killer in the world, and between 30 and 40% of all deaths due to antimicrobial resistant bugs are due to drug-resistant TB. And do we not track it anymore because we just know that it's endemic and it's going to kill a ton of people and we've given up on them? Or what's the scenario from your perspective of pretty much any of your work, but I think maybe especially Jim?
Well, so our bias here is we look at abrupt shifts in indicators that show us that a community is suddenly experiencing something that's acutely happening. And in the tuberculosis world, like with HIV, malaria, and some of these other endemic diseases in endemic areas of the world, we see very calm disruption patterns, right? So these are communities that are used to dealing with these issues. They're desensitized, even though there's a lot of mortality. What we do see are cases where, and we know it's just the tip of the iceberg, I mean, we have data problems just like everybody else does, but where an XDR TB will show up in a clinic in South Africa and people will realize that, oh my gosh, we're dealing with a patient that can't be treated, and then people evacuate the facility, potentially spreading that into the community. And so we've detected that periodically. We know there's a lot that we don't detect, and yeah, it's a huge issue. I mean, we've all discussed, the issues of access to data, which kind of drives everything and drives our awareness. But the classic mantra in public health is if you don't report it, it doesn't exist. Yeah. So.
And I would just add that there are significant pushes to end TB and malaria. And HIV, and we've been talking about explosive epidemic events, but actually there are these very long-term endemic infections that cause a huge amount of morbidity and mortality and continue to place enormous economic burden on low and middle income settings. Those movements with genomics and all of the methods that we've been talking about are hard for these infections that have long chronic latent periods. But there are certainly, there's certainly a whole range of people who are trying to push for ultimately eradication of those things. We've only managed to eradicate 2 pathogens. We're struggling to do so for the third, polio. But that is definitely on the agenda, I think, for most people. It's a really tough challenge, though. I don't know if, Jess, you want to talk about TB, because...
I was just going to mention the genomics as well. So although it's very hard to detect TB through case detection, just because it is this chronic pathogen, we can trace back by looking at the genomics and try and understand patterns of spread, and that again speaks to healthcare capacity and where you need reinforcements. So it is a hidden epidemic, but it's one that we are using the lens of things like, to use the big data buzzword again, is being used to approach these challenges.
Okay, we are getting flashing red lights. I'd like to thank all the speakers once again.